So here at HiSim, I work in the group of Andreas Trump, and we've always been interested in quiescence and dormancy. So features of stem cells that allow them to not take part in steady state everyday blood production, but maintain essentially sleeping in the bone marrow, and then to be reactivated in cases of dire need for blood or immune cells, essentially infections or blood loss. And this is a very unique feature in stem cell systems. For example, in the gut, stem cells are usually not considered to be quiescent and is very tightly regulated by intra and extracellular mechanisms. And we are trying to elucidate them. And I'm going to present one new pathway that maintains stem cell quiescence in the haematopoietic system. The hypothesis is that this process of maintaining stem cells in quiescence is essentially is really essential for keeping stem cells alive during the whole lifetime of a mouse. And if stem cells are not kept in quiescence, they will be functionally inferior. Additionally, we focus on the question whether the mechanisms that maintain stemness during the normal life, during the early lifespan of a mouse or a human being, are altered in aged people, and therefore can explain why the immune system of older people or mice are worse off than the ones of young animals. We used mostly transgenic mice, and RNA sequencing to identify our targets, and then transgenic mice to functionally study whether the molecules that we find in our screenings are relevant. We identified a receptor, it's called neogenin, which is specifically expressed on haematopoietic stem cells and really on the most quiescent haematopoietic stem cells. If you look further to the progenitor cells, which are limited in their functional capabilities, this receptor is not expressed anymore. Therefore, we were quite excited that there might be a unique molecule that is only expressed on the most potent stem cells. And we're wondering about the different ligands, in this case, netrins and repulsive guidance molecules, molecules that have originally been identified in brain biology. So those molecules usually regulate the sprouting of neurons throughout the brain. And now we find this molecular signaling hub in hematopoietic stem cells. And then we started to use plenty of different transgenic mice that allow us to overexpress or knock out netrins and neogenin as their receptor in the hematopoietic system and in the microenvironment surrounding hematopoietic stem cells. So we find that if we knock out either netrin, so the ligand of neogenin, or neogenin, the functional capabilities of hematopoietic stem cells are impaired. That is the first big important thing. And the second thing is that they lose their quiescence. So they get activated more, they cycle more. And if you then transplant them into secondary recipients, they cannot engraft as good as normal non-transgenic hematopoietic stem cells in either system. Additionally, we find that if we culture stem cells in vitro and we stimulate them additionally with netrin-1, so the ligand of neogenin, we can increase the engraftment capability of cultured hematopoietic stem cells, which is an important point considering transplant settings. So one last important point is that we find, if we look at aged mice, then we see that normal aging, the normal process of physiological aging, is deregulating, disrupting the expression of neogenin ligands, especially netrins. And we think that this, in combination with the disruption of the expression of other molecules that are essential for a hematopoietic stem cell maintenance, like stem cell factor, for example, CXCL12, is an important factor of hematopoietic stem cell dysfunction in aged patients. And for netrin, it's similar. So therefore, we think that we can contribute to the biology of hematopoietic stem cell aging in a way, and that we think that aging is more an extrinsic process. So the microenvironment is changing, and that changes the hematopoietic stem cells.